So I have two neurons that are going to be communicating with one another. And you guys know enough anatomy to look at two neurons in series and tell me which direction the information is flowing. Do you not? Especially if they're multipolar neurons like this, which is what you're used to looking at. So I know you know, but tell me, which direction is information flowing between those two? Right? It's flowing down. That means that for any given synapse, you can tell me that one of those neurons could be considered the presynaptic neuron and one of them could be considered the postsynaptic neuron, correct? Right? Presynaptic and postsynaptic. And so what we are going to do is to talk about the synapses. Today, we're going to be talking primarily about what we call a chemical synapse, which means you are going to release chemical messengers to communicate between the two cells. There is also another kind of synapse, but it doesn't really exist in the adult nervous system. It's called an electrical synapse. An electrical synapse would be when the presynaptic and the postsynaptic cell were stuck together via gap junctions. And then whatever happened to this one, depolarization or hyperpolarization, would happen to that one. And that's all well and good, and it would be super duper efficient, I think we could agree, because it would almost allow for a continuous action potential between them. But that would mean that cell A and cell B would always have to do the same thing. Do you really want all of your neurons turned on simultaneously in your brain? No, that would make crazy or dead. I don't actually know which one. But yes, you need to be able to turn some on and some off. And by pulling them apart, right, you can have this postsynaptic one have receptors for a bunch of different things, so it can respond differently than the presynaptic neuron. Okay, so we do have electrical synapses like in cardiac muscle between the cardiac muscle cells, right? Other places as well, but they're relatively rare in adults. So what we're going to be talking about is what we call true synapses, and it's a neuron-to-neuron communication. And then the next chapter we'll talk about neuron-to-muscle communication, which will be called a neuromuscular junction. But here, assume that in this set of notes, unless I tell you otherwise, we're always talking about two neurons communicating with one another. And so what we have to get down is just sort of the basic functional anatomy of how these chemical synapses work. So what I want to use is a pitcher, catcher, ball, mitt analogy. So when we are looking at the relationship between these two neurons, first we have to get down the basic anatomical terminology. And unfortunately, your physiology book and your anatomy book don't use the same terminology a couple times, so let's make sure that we all know what we're talking about. So in the presynaptic cell, we're going to have the cell body, and we're going to have the axon, right? And then at the end of the axon, we have fingers and we have fingertips. Does anybody remember what the fingers are called, which you can see? So the ET fingers that they have. Let's see, I'll find a branched one so I can show you. Okay. So do you see how this one branches into multiple fingers? So what are those called? No, not the axon terminals. The axon terminals are the tips. Telodendria, which literally means fingers at the end. So I have my axon telodendria. There's no limit to how many telodendria you can have. And at the tip of each one of them, it's kind of a bulbous end to the telodendria. And that is called, depending on whether you look at your anatomy textbook or your physiology textbook, a synaptic terminal or an axon terminal or a synaptic knob. It's all the same thing. Why is it bulbous? Why does it look like it does at the end? What do you have stored there? What is stored in the axon terminals? Yeah, that's where the neurotransmitter is stored, exactly. Okay, so our pitcher-catcher-ball analogy that we're going to be working with here, let's get the players down. So when we're dealing with the players in the pitcher-catcher-ball analogy, the presynaptic cell will be the pitcher, yes? The postsynaptic cell will be the catcher, and the catcher needs a mitt, so what's going to be the mitt? The receptor on the postsynaptic cell membrane, right? And then what's going to be the ball? Well, ultimately what you're going to do is the neurotransmitter is going to be released into the synapse from the pre to the post. And then it can cause a response on the postsynaptic cell because it binds to the receptor. Does that make sense? So you've got your pitcher, your catcher, your ball, and your mitt. Now notice that in this particular metaphor, the pitcher doesn't have a mitt. That means that even if I sent the neurotransmitter back across the synapse, the presynaptic cell will not respond to it because it doesn't have a receptor for it. Does that make sense to you? So because of this anatomy, that means synaptic transmission is one way, right? That means if you see the two, you know which direction the information is going, and you can cause a response from pre to post. But even if I sent it back, there's no receptor, so I'm not going to be able to cause a response backwards. Does that make sense? So imagine that in this analogy, the pitcher can throw the ball and the catcher can catch the ball, but the catcher cannot throw it back. He has to roll it back, right? So for instance, the presynaptic cell can reuptake, but it's not going to respond to the neurotransmitter. Okay, so our players, the functional anatomy of a chemical synapse, the axon terminal is the swelling at the end of the axon, also called the terminal boutons, or there's a bunch of different names for it. The synaptic vesicles are where the neurotransmitter is stored. Generally, although I don't know if it's always, the neurotransmitter would have been made up in the cell body and then transported down to be stored at the end of the axon, and you store it in vesicles, which means it's going to be released by exocytosis. So we will release this by exocytosis. And we used to say that a single neuron makes, stores, and releases a single neurotransmitter. But just recently they found out that there's actually an exception to that. There are some neurons that make, store, and release more than one neurotransmitter. But I have no idea how they do that, and I don't think we actually know yet. So we're going to hold it to, if this was, for instance, acetylcholine that was stored in those synaptic terminals, then let's just say that this one makes, stores, and releases acetylcholine. And eventually we'll figure out how that other thing works. So you've got your synaptic vesicles, you've got your presynaptic and your postsynaptic, and they are separated by this space, which is a synaptic cleft. What should be in the synaptic cleft? What's there? Yeah, it's fluid. It's extracellular fluid, correct? It would be interstitial fluid to be more specific. So the synaptic cleft is actually this physical space between the two of them. And although if you look, it looks almost like these guys are touching, they're not touching. They're microscopically separated, okay? There is room for things to be released between the presynaptic and the postsynaptic cell. So that's the synaptic cleft. The postsynaptic neuron is going to be the one with the receiver, with the receptor. And the neurotransmitter is going to be the ball in this metaphor. So I know I told you, but now I want one of you guys to tell me, why does this picture tell you that synaptic transmission is exclusively one-way transmission? Go ahead. Right, so even if I send it back, you won't get any response to it, right? That means that the action is all happening one way. Okay, so now, let's start looking at the functionality of this guy. So now we're going to add a little bit more detail. And there is a type of receptor that I haven't introduced you to yet. So the part of the story that we already told, and we told in the last set of notes, started with the presynaptic cell. Okay? So the presynaptic cell, we said that basically we're trying to get an action potential. So if the cell body is down here, the cell body, or the dendrites, could, for instance, respond to, let's say you flooded it with acetylcholine, that bound to and opened acetylcholine-gated sodium channels, right? Does that make sense? Chemical-gated sodium channels? Yes, chemical, and acetylcholine is the chemical. You guys with me? Okay. You open chemical-gated sodium channels. If you get a big enough depolarization that the axon hillock hits -55, then what happens? You get an action potential, because you're going to open the first voltage-gated channel and then the second, and then you get all the way to the end. But what we didn't tell is what happens when you get to the end of the cell. When you get all the way to the end of the axon, you're going to open a new kind of channel, and this new kind of channel is right here. It is a voltage-gated calcium channel, and it opens in response to depolarization, but it's only found in the axon terminals. So isn't it going to be the last thing that gets depolarized? Yeah? Axon terminals are going to be at the very end. You're going to open the voltage-gated calcium channels, and then calcium is going to move down its electrochemical gradient. It's higher on the outside, lower on the inside, and of course the inside is negatively charged, right? Who cares if calcium moves in? It is the increase in concentration of calcium that is the trigger for exocytosis. So as calcium moves in, then the vesicles start moving to the cell membrane, and then you do exocytosis of whatever neurotransmitter is there. So let me show you an animation, give you a visual of this. Okay, so presynaptic, postsynaptic cell, they are just representing the action potential as a zap, right? Then you open your voltage-gated calcium channels. Calcium rushes in. That triggers exocytosis of the neurotransmitter. In this instance, the neurotransmitter is acetylcholine, which is going to bind to and open chemical or ligand-gated sodium channels, what is going to be happening on this postsynaptic cell? What kind of polarization change? D or hyper? Yeah, that is going to be depolarizing the postsynaptic cell. Does that make sense? So the story that I'm telling here is that if I got an action potential on the presynaptic cell that causes neurotransmitter release at the end, then I'm going to release that chemical, neurotransmitter, which will bind to chemical-gated channels on the next cell, right? So it's the same story that you restart over and over again. Does that make sense to you guys? Okay, so let's look back at your notes. So the neuron A and neuron B, or the presynaptic and the postsynaptic, are separated by a synaptic cleft. And this is always hard to explain, but see if you can conceptualize this. You know what an action potential is, right? It's a big wave of depolarization that moves down a cell membrane, right? Caused by ions moving across the cell membrane. Is it possible to have a continuous action potential if you have cell membrane and then no cell membrane and then the cell membrane starts again? You can't have a continuous action potential. So what happens is, as long as there's a cleft between the two of them, then you have to restart the whole story every time. Does that make sense? So we're going to restart the same story that we started down there. I started with a whole bunch of chemical, right, binding to chemical-gated sodium channels. If I get depolarized enough, I get an action potential. So what I'm saying is, at the end of this story, that's the beginning of the next story, right? That's the beginning of the next graded potential that could lead to an action potential. So I got to the end, I opened my voltage-gated calcium channels, calcium rushes in, causes exocytosis of the neurotransmitter, and now I have a pile of acetylcholine that is going to bind to chemical-gated sodium channels, potentially, on the next cell. If I get depolarized enough, then I will depolarize that axon hill up to threshold, and then I'll get another action potential. So the truth is that you restart the story every single time. It's not a continuous story, it's a repetitive story. Does that make sense? Okay, so the space is bridged with chemical messengers, and the chemical messengers in this particular story are what kind? What are they? Acetylcholine is specific, but what are they generally? They're neurotransmitters. But let me back up and make you tell me what 2 criteria you have to meet to be a neurotransmitter. What are the 2 criteria that you have to meet to be a neurotransmitter? Released by a neuron and? Nope, you don't have to communicate with a neuron. You have to be released right next to the receptor, meaning that I can't dump it into the bloodstream. So do you see that this meets those criteria? Released by a neuron right next to the receptor? Okay, so whatever neurotransmitter you had is the one that you released, and then the neurotransmitter is going to bind to the receptors on the postsynaptic cell membrane. So what we're going to talk about now is what can you do to the postsynaptic cell. So we just said that you could get it to have an action potential, but we know that you do not turn all your neurons on. We have the capacity to turn them off as well. So now let's talk about how that occurs. So what's going to happen is the neurotransmitter is going to be released, and it's going to bind to whatever kind of receptors there. So let's talk about some kinds of receptors. So if the neurotransmitter is released and binds to a chemical-gated sodium channel, for instance, okay, just ignore potassium in this one, they're trying to confuse you. So binds to a chemical-gated sodium channel, then it's going to depolarize the postsynaptic cell. Everybody with me so far? If you had to choose, would you say that depolarization would represent excitation or inhibition of the postsynaptic cell? Meaning, am I getting closer to or further away from an action potential? Closer to, choose the word. Excitation, exactly. So what I'm doing if I bind to and open chemical-gated sodium channels is I am actually causing what your textbook calls an EPSP. Now, it sounds confusing, but it's really not. Excitatory, because it is. Yes, it's depolarizing. Postsynaptic, because that's the cell you're messing with. Potential, because it's a membrane potential change. Does that make sense? So if I said that in this particular instance that we were talking about, this neurotransmitter bound to and opened a chemical-gated sodium channel, then I could tell you that this entire synapse was excitatory and that what happened on the postsynaptic cell was an EPSP, excitatory postsynaptic potential. Does that make sense? Now, does that guarantee that you're going to get an action potential? No, it just went the right direction for an action potential, right? It might go from minus 70 to minus 68, but it's still the right direction. Does that make sense? So you could say, in this instance, the entire synapse is excitatory. It's causing an EPSP. And you could say the neurotransmitter was excitatory at this synapse. Does that make sense? Okay, so the kind of receptor that does this one, in which the receptor is an ion channel. These are called ionotropic receptors, and they're the fastest ones that we have. The receptor is also an ion channel, so this is an ionotropic receptor, but it doesn't have to be a sodium one. What if different neurotransmitter, for instance, bound to and opened a chemical gated potassium channel? What kind of polarizing change would that cause on the membrane? Hyperpolarizing. And if you had to match that to excitatory or inhibitory, what would you say? Inhibitory, right? Because it's getting further away from minus 55. That means that in this particular instance, if we use the same figure and we say, okay, now we are binding to and opening a chemical gated potassium channel, then this would be an inhibitory synapse, and your textbook would describe what is happening on the postsynaptic cell as an IPSP. Inhibitory, yes, because it's hyperpolarizing. Postsynaptic, that's where it occurs. Potential, because it's a membrane potential. Does that make sense? So we've got EPSPs and IPSPs. So can you understand that? With that in mind, if I released a neurotransmitter that was inhibitory, for all practical purposes, I would be turning off or down the postsynaptic cell and making it less likely that I'll get an action potential. Yes? But if I release something that opened a sodium channel, I would be turning on or making it more likely. And all of that is happening in your brain because you do not want all of your neurons to be on simultaneously. So you're actively inhibiting some of them and actively exciting others. Does that make sense? Okay, and it's different ones depending on what kind of activity you're doing. So conceptually, I'm going to back up just a little bit. You don't have to use sodium for excitation, right? It's just the most common one. Anything that depolarized the neuron, whether it's calcium or sodium or chloride moving the other way, would have this effect. Same thing with hyperpolarization. You don't have to use potassium for hyperpolarization, but it's the one that makes the most sense because of our sodium-potassium pump setting up the concentration gradient. Okay, so concept check. This is kind of an interesting thing when I figured this out. So epilepsy is a condition in which for whatever reason, maybe injury, maybe genetics, whatever, your neurons fire inappropriate action potentials that aren't supposed to be happening. And it can cause like a big grand mal seizure in your brain, which could cause muscle contraction as well, but doesn't have to. So you've got inappropriate action potentials. Does it make sense to you that one of the first tier of drugs that they use to treat epilepsy is a drug that hyperpolarizes your neurons? Does that make conceptual sense to you? Because it makes it less likely that you'll get inappropriate action potentials, but it also makes it less likely that you'll get appropriate action potentials. So compliance with that particular drug is a little hard because you have a bunch of seizures and then you go on the drug and then you feel mentally and physically lethargic, but you're not having any seizures, and then you go off the drug, right? And you feel like you're with it again, and then you start having seizures again. Does that make sense? There's a bunch of different drugs that treat epilepsy. The one that I know that hyperpolarizes the neurons is called primidone. Okay. Do you have a question? No? Okay. So there is another type of receptor, and we're only going to briefly talk about this one. We will talk about it in more detail when we get to the endocrine system. But I want to compare these two just so that you can know that this occurs. This one right here is the one we've been talking about. The receptor is an ion channel, and this is called an ionotropic receptor. And they tend to be pretty fast. There's not a whole lot that has to happen. You open it, it depolarizes or hyperpolarizes. But there is one that's way more complex and also slower. And this is called a metabotropic receptor. And the metaphor that works for me about these is this is like a direct shot in pool, okay? The pocket's right there, the cue ball's right here, and the ball that you want to hit is right here, and you go straight at the pocket. But you know some people like to do super fancy combo shots and go through 4 balls to try to get there. That's what happens with this one. So look, You don't have to know the details later. Just conceptualize or right now, just conceptualize what I'm talking about. Neurotransmitter binds to the receptor, but the receptor is not the ion channel. The receptor is ultimately going to cause the ion channel to open or close, but it's going to do it by involving 5 of its closest friends to get there. These are called G protein receptors. They use these 4 little proteins that are powered by guanosine triphosphate, or GTP. And ultimately what they're going to end up doing is just opening this freaking channel just like you wanted to do the first place. But these are way slower receptors. They still have the same activity. They're still either going to depolarize or hyperpolarize the cell membrane. Okay, so we are not going to study these in detail yet until we actually get to the endocrine system. I just want you to know that they exist. Okay, so the ultimate function here was that we were trying to cause a new graded potential on the next cell, either a depolarizing one or a hyperpolarizing one. And let's say it's depolarizing. If you are depolarizing the next cell, you could get an action potential in the next cell, yeah? But one last thing. Remember that with nervous system, we use frequency coding for intensity. Does it make sense to you that if I released a neurotransmitter and then left it in the synapse, I would, for all practical purposes, be insensitive to the next release of neurotransmitter? Does that make sense? If I just left it there? So what I have to do is I have to release it into the synapse, let it do its thing, and then I have to remove it from the synapse so that I can respond to the next release. So with neurotransmitters, you have to dump them into the synapse and then you have to get rid of them very quickly. And there's several mechanisms that you can use to get rid of neurotransmitters in the synapse. One of them is just letting them diffuse away. But that's not a very effective reset. So it's not going to make the cell really responsive to the next release of neurotransmitter. So you could wait for them to diffuse away. But quite often there are a couple of other removal mechanisms. One of them is this. Quite often on the postsynaptic cell membrane, there is an enzyme which will degrade or change the shape of the neurotransmitter so that it won't cause another cell response. And really all you have to do is change the shape just a little bit, right? So for instance, with acetylcholine, one of the most common ways to remove it from the synapse is to use an enzyme called acetylcholine esterase. And what it does is takes acetylcholine, right? Breaks acetylcholine into acetic acid and choline, okay? And then it can no longer cause a postsynaptic cell response. And because your cells are great recyclers, they take the choline back up and make more acetylcholine out of it and then do it again, right? So this is another mechanism of removing it from the synapse with an enzyme in the postsynaptic cell. And then there is another common mechanism, and we'll talk about this one in a decent amount of detail before we get done. And that is that the presynaptic cell reuptakes the entire neurotransmitter, right? So I released it and I reuptake it. And this reuptake mechanism will just allow for it to get repackaged and then I can release it again. Note to self, we said that this will not cause a presynaptic cell response. It's just a reuptake mechanism. And then the last one that I didn't even know until a couple of years ago is they just figured out recently, or at least I just learned recently, that there's one other way that you can get them out of the synapse. Remember our friends, the glial cells or neuroglial cells? Apparently, the glial cells sometimes reach into the synapse, grab the neurotransmitter, and hand it back to the presynaptic cell. So they're actually helping with this removal and recycling process, which is pretty cool. They're finding out more and more that glial cells are super important players in the nervous system. Okay, so that's the primary mechanisms that you can use to get rid of these things. So let's talk about that reuptake mechanism again. So the reuptake mechanism is something that we know a decent amount about because some of our favorite chemicals are dopamine and serotonin. I always love this little picture. It says serotonin and dopamine, they're technically the only 2 things that you enjoy because those are your enjoying chemicals, but I would add one more, which is norepinephrine. Norepinephrine is kind of alert, and we'll learn these in more detail. Dopamine is a reward neurotransmitter, and serotonin is kind of your leveling neurotransmitter. And the reason we know a decent amount about these is because when people have disturbances of, for instance, serotonin, if you have low levels of serotonin that are being released in certain portions of your brain, it can manifest as depression or anxiety. And in theory, what you should do is increase the amount of serotonin. But conceptually, it's pretty hard to make cells make dependable amounts of serotonin just because you want them to. So what they do instead, quite often, is inhibit the reuptake of serotonin. So if in this figure, where were we? If in this figure we say this is serotonin, okay, and I'm not making enough serotonin, does it make sense that the postsynaptic cell would behave as if there were more frequent serotonin release if you just left the serotonin in the synapse for longer, right? And so an entire class of antidepressant and anti-anxiety drugs, and they're kind of the first tier of treatment for antidepression and anti-anxiety, is SSRIs. They are selective serotonin reuptake inhibitors. And what they do is at selective synapses, preferably just the ones that are related to mood, they will inhibit the reuptake of serotonin. Does that make sense? And you will have all of the manifestations of more serotonin in your system. Now, why do they have to be selective? Because serotonin doesn't just do mood. It also does hunger and sleep and sex drive and hallucinations. So if you are not super selective, you're going to have all kinds of unintended side effects. That makes sense? So that's the reuptake mechanism, and we'll talk more about that. Okay, so now, what we have sort of been telling is we've sort of been implying that the story is one pre to one post, but that's not generally true. So in the nervous system, it's usually not a one-to-one relationship between pre and post. As a matter of fact, the central nervous system is more like 15,000 connections for every neuron. But you can understand why I don't try to teach you 15,000 connections before I teach you one, right? You'd all drop the class. So what I have here is a bunch-to-one relationship. And so there are two ways to think about this. So the relationship of presynaptic to postsynaptic cell is very seldom one-to-one. Like I said, it can be as many as 15,000 to 1. So there are two ways to look at this. The first way is convergence, and the second way is divergence. So you know what those terms mean, right? If I say you're converging on something, there's a bunch of things that are coming into one little point, right? And if you're diverging, you're starting out at a point and you're going out. Okay, so with that in mind, can you see that this picture with a whole bunch of presynaptic cells converging on one postsynaptic cell, right, is an example of convergence? Okay, now what is it trying to tell you? Why are they different colors here? Because they're trying to represent that some of these synapses are going to be inhibitory, the red ones, stop. Some of them are going to be excitatory, the green ones, go. Does that make sense? So what is this cell going to do? Is it going to be excited or inhibited? Whatever happens at the axon hillock, if it gets to minus 55, it has an action potential, and if it doesn't, it doesn't. Is that okay? Okay, so that's convergence. That's the idea of convergence. And here's the idea of divergence. I've got a single cell, and yes, it only has one axon, but there is no limit to how many times the axon can branch, right? So this is one cell that is going to branch and diverge and form synapses with multiple other cells. So for all practical purposes, does it make sense to you that these 4 cells would therefore act kind of like a team? Right? Because if you get an action potential, they're all, not necessarily simultaneously, but at least around the same time, they're all going to respond to the same neurotransmitter, right? You're going to run into this idea of divergence quite often in the muscular system because one neuron can control up to 500 skeletal muscle cells. And they behave as a single unit, and it's called a motor unit. Okay? All right, so that's the idea of convergence and divergence. So what the axon hillock does in the postsynaptic cell is it is actually going to, for instance, right here, this axon hillock, it's going to integrate all of those different inputs. And if some of them are inhibitory and hyperpolarizing and some of them are excitatory and depolarizing, the only thing that matters is whether the axon hillock gets to minus 55, right? So you can depolarize, you can hyperpolarize, and the axon hillock doesn't care about any of that part of the story. If it gets to minus 55, it has an action potential, and if it doesn't, it doesn't. Okay, so they call that integrating. The axon hillock will integrate. Okay, so interesting little tidbit. So synapses. Synapses are these connections you have between one cell and the other. There is a tremendous amount of new information that says that there is a relationship between learning and synapse formation, and that synapse formation primarily happens when you're sleeping. So it's not as if one synapse equals a new memory, but what is actually happening in your brain when you're learning new things is, like I said, there's no limitations to the number of times the axon can form a branch. So if I learn something, I actually form branches off of the axon and actually make new physical connections with new places in the brain, which is kind of fascinating because we have been using that description of making connections when you learn things for much longer than we actually knew that there were connections being made. And it seems as if a large part of the connection making happens during sleep. And so the neurologist that I listened to about this, I listened to a podcast, this neurology, and the metaphor that he used was like when you are awake and all of your sensory receptors are on, it's like you're being bombarded with new information and you're just sort of taking it in, but you don't organize it. The organization, the sort of making connections and putting things together seems to happen when you're sleeping. And so that's one of the hypotheses as to why your brain has such a high metabolic rate when you're sleeping, when it's not actually taking in any sensory information to speak of. And one of the reasons that they started thinking about this is because dementia and Alzheimer's and other forms of dementia, they quite often manifest as sleep disturbances long before they manifest as memory disturbances. And so the hypothesis existed long before they could confirm it that there was some relationship between sleep and memory. And so they're starting to get reinforcements that that's probably the case. Okay, so now this postsynaptic cell. So we're saying this postsynaptic cell right here. We want to talk about how we can figure out whether it's going to get an action potential or not. Okay, so with the postsynaptic cell, what is not generally true is if you've got two cells, let's just say there were two, pre and post. One action potential on the pre does not usually cause an action potential on the post. What has to happen usually is you're going to have more than one action potential on the pre to get enough neurotransmitter release so that you get the post to an action potential. Okay, so the many action potentials are generally required. This concept is summation, and this means that the whole process really slows down in the synapse, right? So it goes 250 miles an hour down an axon, but then if you have to do that multiple times, that means that the synapse is going to be the slowest part of the nervous system. So they call that concept that you actually slow down at the synapse, they call this synaptic delay. And the synaptic delay is usually anywhere from 0.2 of a millisecond to 5 milliseconds. But you have no capacity to notice that because your noticers work with that synaptic delay. So there's no way you can really notice it, right? But it is the slowest part of the nervous system. So let's talk about how you can get that postsynaptic cell to an action potential, and there's a couple of different ways. So let me orient you to what this picture is looking at before we go any further on it. Okay, so I want you to look. Here's my postsynaptic cell, and that's what I am trying to get to threshold. And really, I'm only concerned about the axon hillock getting to threshold, right? So the little voltmeter is set at the axon hillock. And I've got 3 synapses that I'm going to be dealing with. I've got A, B, and C. And they are trying to color code it to represent A and B as excitatory synapses and C as inhibitory synapses, right? So the question I'm trying to answer is how do I get that postsynaptic cell to an action potential? And there's really 2 different ways of accomplishing it. If we just look at the top picture right here and only concentrate on a single synapse, let's call it A. Let's just look at A. So if I stimulate A, and A is, let's say, a chemical gated sodium channel, right? I open that chemical gated sodium channel, I get a little bit of depolarization, right? Not enough to get me an action potential. And then I let it completely repolarize back to -70 and then I stimulate A again. I'm still not going to get an action potential. But what if, instead of going zap, pause, zap, what if I go zzz? What's going to happen is I am going to add to the number of open chemical gated sodium channels, right? And I'm going to add the depolarizing effect of opening more than one of them, right? So what I'm going to do is instead of stimulating A infrequently, I'm going to stimulate A frequently, right? Does that make sense to you guys? So there's more, for instance, acetylcholine around opening more chemical gated channels. and getting me to threshold. Does that make sense? Okay, so this concept is called temporal summation. And temporal, what does that mean besides the muscle and the bone? What does it mean? Time, exactly. So this is a frequency summation kind of thing. It means if you stimulate the presynaptic cell more frequently, you can get the postsynaptic cell to an action potential. Yes? Now, the question that comes up for people, I want you to listen to this because if you think about it at home, you can confuse yourself. So what people do right here is they think, well, what about the refractory period? But I want you guys to remember that the refractory period does not occur between minus 70 and minus 55. The refractory period is only after you open the voltage-gated channels, right? So the refractory period occurs here. There is no refractory period here. You can add those graded potentials together all you want. Does that make sense? The refractory period, remember, has primarily to do with the status of the voltage-gated sodium channels, right? So you can stimulate very frequently. You don't have to wait for it to repolarize at all, as long as you're between minus 70 and minus 55. Is that okay? So this is temporal summation. It only requires one pre, one post, okay? But there's another way to do it. And the other way to do it is you can use multiple pre to a single post. And this is called spatial summation because I'm going in at more than one space or more than one place. So if I stimulate A alone, it's depolarizing, right? I get excitation. If I stimulate B alone, I get excitation. If I stimulate C alone, I get inhibition, right? But what if I stimulate A and B simultaneously? Two different synapses, both of which are depolarizing. Can you see that I could actually get the cell to threshold by doing that as well, right? That's not frequency summation. That is spatial summation. It's two different synapses. Does that make sense to you guys? Okay, now, A and B simultaneously. Now, can you sum to get to 0, or could you sum and stay at minus 70? What if I did A and C, for instance? Right? A was depolarizing and C was hyperpolarizing, and I could end up right there. So you can sum and have nothing actually happen to the cell. And that can happen when, for instance, one input wants to start the cell in action potential and the other one wants to inhibit an action potential. What wins? Well, it just depends on whether you get to minus 55 at the axon hillock or not. Doesn't really matter how you got there. Okay, so this is spatial summation because it's more than one space, and this is temporal summation. So my question to you is, which of those requires convergence, spatial or temporal? Spatial requires convergence. Temporal does not. Does that make conceptual sense to you? Okay. So that's temporal and spatial summation. Okay, questions? Any questions? No? Go ahead.
So what would cause it to have both the inhibiting and the excitatory at the same time?
Oh, because, so here, think about this. Let's just imagine that this postsynaptic cell is a hypothalamus cell, okay? And the hypothalamus gets input from light and temperature and metabolism, right? Does that make sense? It's gathering information from a bunch of different places. And some of them may be saying, I want you to have an action potential. And the others may be saying, no, I don't want you to have an action potential. So all of the cells that are presynaptic to this cell don't necessarily have the same agenda for the cells. So some may be starting it and some may be stopping it. If you want to think about conceptually, when you are trying to make yourself get out of bed in the morning because your alarm rang, but you didn't get any freaking sleep. So there is a part of your brain that says, go back to sleep. You need to sleep. You're an idiot. And the other part says, get up. You have a job and you want to keep it, right? And so you've got these different parts. Which one wins, right? Whichever one is most persistent, really, is the one that wins. Do you need sleep or a job? Which one do you need most? So it's whichever one is most persistent. And sometimes you don't have necessarily the same agenda for all of the cells, right? And also changing activities, right? You were doing one thing and you're going to move to doing the other. So you can imagine that with the cell in that situation. Okay, so one last question for you about this, and then we'll change gears a little bit. What do you think is the absolute fastest way to get that postsynaptic cell to an action potential? What? Spatial? How about you do both of them? You do spatial summation very, very frequently, right? You do a whole bunch of presynaptic cells very, very frequently, and you can get that postsynaptic cell to an action potential more quickly that way. Does that make sense? Okay, all right. So let's say that you are not releasing adequate amounts of whatever neurotransmitter you're dealing with. So it doesn't really matter which one you're going to talk about. So you're releasing a neurotransmitter, but you're releasing too little of it to have normal physiology. So let's say this particular synapse right here is a serotonin synapse, and I'm not releasing adequate serotonin. So what can you do? Well, not surprisingly, because the synapse is actually accessible and by the extracellular fluid, a lot of the medications that we use will actually affect the synapse, right? Does that make sense? So we use medications that don't necessarily, they're not as likely to affect the intracellular like cell functionality as they are to affect what's going on between the cells, the communication between the cells. So in theory, if I had too little serotonin, like I said, you'd just say, well, make more serotonin, right? Just make more. And we don't really have any way of doing that in a human, because if I said make more serotonin, I'd have to say, but just in these cells and not in those cells. And then you'd actually have to change the intracellular activity of a cell. So in theory, if you're making too little serotonin and you are depressed, just make more. And there are some things that you can do to make yourself make more serotonin. You can get some more sleep, exercise more, have more sex, and eat better. But they are not going to cause a dependable clinical manifestation. So a lot of times what they do is skip to the next thing. And the next thing is, if you can't make yourself make more, make the stuff that's in the synapse stay there for longer. So if you release it, you let it sit in the synapse for longer, like the SSRIs that we were talking about. So you can interfere with it being removed from the synapse. An SSRI, a selective serotonin reuptake inhibitor, would block this part. And then your postsynaptic cell would, for all practical purposes, think you had more serotonin. Or what about an enzyme? If there was an enzyme that broke it down, you could block the activity of the enzyme. Does that make sense? Okay, so a lot of our antidepressant and anti-anxiety drugs will do exactly that. And specifically with these lovely chemicals, which are your favorite chemicals, serotonin and dopamine, but really it's actually these three. Serotonin, dopamine, and norepinephrine are the three that we most commonly manipulate to treat depression and anxiety. And norepinephrine is your up one, dopamine is your reward one, and serotonin is sort of your level one. And if you've got perfect amounts of all three, you are going to be in this perfect mood. Pretty sure I've been there for like 5 minutes in my entire life, right? So one of them is probably always out of whack. So we'll talk about those three in a lot of detail. So you could interfere with removal of the neurotransmitter from the synapse if you had too little of it. And then here's another fun way to think about. So a lot of what we do clinically is if we can't make you make the right amount, and we're not going to do what we just said, how about we interfere with what's going on with the receptor? If we can make a drug that looked just like the neurotransmitter, then you could get a relief from too little of it. And if we could make a drug that blocked the activity of the neurotransmitter, you could get a relief from too much of it. Does that make sense? So there's two categories of drug that fit in there. They are called agonist drugs or chemicals and antagonist drugs or chemicals. If you are an agonist drug, you fit into the receptor and cause a response that is similar to your naturally occurring chemical, right? It's a mimic. That's how I think about agonists. And with agonists, here's an interesting one. They produce a response that's similar to the normal activation of the receptor. It's usually not identical, because anything you make externally, it's usually not completely the same as what you made internally. Nicotine, fascinatingly, part of the reason that people like nicotine when they first start using nicotine is because nicotine is an acetylcholine agonist in your brain. And acetylcholine, one of the functions and part of your brain, is that it increases alertness and increases your short-term memory. So you feel kind of smart for a second. You feel kind of with it, right? So I know everybody says, don't smoke, don't do drugs, but I always think it's really useful to understand what's the appeal in the first place, right? Why would you start doing something like that? Well, it makes you feel kind of with it. And then, of course, you downregulate your receptors and it doesn't work anymore and you're just kind of addicted to it. So nicotine is an acetylcholine agonist. and it impacts these parts of your brain that make you increase your short-term memory and increase your sort of alertness. Antagonist drugs or chemicals would sit in the receptor but not cause the response. And usually they hold on to the receptor kind of tight, otherwise they wouldn't really be useful for very long periods of time. So these are called antagonists, and I think of agonists as mimics and antagonists as blockers. Does that make sense? So here's a fascinating one. One of my favorite chemicals is caffeine. See if you can conceptualize this. Caffeine is an antagonist to an inhibitory neurotransmitter. Can you think about why it has the effect that it has? It's an antagonist to an inhibitory neurotransmitter. So if you block the inhibitory response, then what do you get?
Excitation.
You get excitation, exactly. That's how caffeine works. That's how it works on your brain. It does other things in different places in your body. So that's how you get the excitatory response of caffeine. It's an antagonist to adenosine, which is an inhibitory CNS neurotransmitter. That makes sense to you guys? Okay. And then, not surprisingly, if antagonists bind to the receptor, don't cause the response, and quite often hold on very tightly, does it surprise you at all that a lot of poisons are antagonists? Right? If you block the response and hold on tight. So let's talk about a couple of these. Tetrodotoxin from pufferfish. Do you guys know that people eat pufferfish, right? Okay, so well, pufferfish are trying to defend themselves from things eating them, and apparently it works pretty well in the sea, but humans are just really persistent. So they make a toxin in glands, and this toxin is a neurotoxin, and what it will do is its antagonist effect blocks a receptor that causes respiratory failure while awake. So you're basically going to be completely awake and aware of the fact that you can't breathe as you die. But humans have decided that fugu, which is the pufferfish prepared as sushi, is absolutely amazing and worth paying hundreds of dollars per plate for. Because apparently has anybody eaten fugu? I've only ever met one person who has. I will totally try it. But apparently when you eat it, it's supposed to be A, delicious, and then B, you get a little tingle, and that tingle means, ooh, you almost died. Yeah, and you can't get it here. I think you can get it in some areas of California. You can, of course, get it in Japan, and people really like it. And people are always like, why do they like that? And my thought about that is I think everybody's just bored after age like 15. You're like, is that all this thing does? I'm totally bored. What am I going to do for the rest of my life? Eat some fugu? Jump out of a plane? I don't know. What are you going to do? So that's fugu. Saxitoxin, which is from red tide. Hey, what is red tide? Is it protozoan? Okay, so, huh? Okay. So I was trying to remember. I was like, I think it's a unicellular. Okay, so in certain environmental conditions, there is a dinoflagellate that has these blooms in the ocean. And it just becomes incredibly common, and it gets into all the seafood, and it's a neurotoxin, and if you eat anything out of the ocean during that time period, you are not unlikely to die. So, and that one's actually an antagonist as well. And then these are two. Black mamba venom is an antagonist, and it just sits in your neurotransmitter receptors, and that's why it kills you. Sea anemone and honeybee venom also are, but those are usually just very local, right? They don't go into your bloodstream. They're not going to cause like an impact on your brain. Okay. So diseases can also do a lot of the things that the drugs can do. So just briefly, let's talk about tetanus and botulism, because your mama told you about both, right? What'd your mama tell you about how you get tetanus?
Step on a nail.
Step on a rusty nail, usually. And she was, as always, partially right. Okay, it doesn't have to be a nail, and it doesn't have to be rusty. It's from this bacterium called Clostridium tetani, which when you get it into anaerobic conditions will produce a toxin. And this toxin actually destroys your ability to release acetylcholine at skeletal muscles, and so it can cause paralysis. Tetanic. Okay. Botulinum toxin, and this is from Clostridium botulinum, same genus, different species. This one, too, is only really problematic when it gets into anaerobic conditions. A really commonly occurring bacterium, but the toxin that it produces when it gets in anaerobic conditions will cause respiratory failure because it's a neurotoxin. But it only does it in anaerobic conditions. So when you can stuff, right? I've only done it once because it was a lot of work for a can of food, really. I was like, last time I checked, I can get that at the grocery store. So you're canning What are you supposed to do to the jar? Boil the hell out of it. What are you supposed to do to the lid? Just completely boil it for way too long to be reasonable. And then you can the thing, and then you put the jar and the lid together, and then you create a vacuum, right? Okay, but the deal is if there were any of these bacteria still alive before you created a vacuum, then they're going to start to produce the toxin in anaerobic conditions. And the toxin is not the thing that you detect. How did your mom tell you that you get botulism? Yeah?
So sorry, I got botulism, and I was in the ICU for 3 months on full life support.
Oh my heavens.
Baby, I got infant botulism. It was really rare. It was airborne before my gut tube was developed enough to fight it away, because once you're over 8 months old, you don't get airborne infant botulism anymore.
Wow, I don't even know that I knew that there was infant botulism airborne.
It's really rare, but there are a few babies in Utah that got it the same year I did. So,
how does it become airborne?
They grow in dry alkaline soil, and so they're released.
Fascinating.
Yeah.
Oh, I'm glad you're here.
Yeah, I made it too, but yeah.
So it is, infants are particularly susceptible to botulism. You don't want to feed them home canned food. But how did your mom tell you that you get botulism, other than airborne spores that I didn't even know about? Dented cans, and she was partially right. It's not the dent or the can, really. It's that if there's a bacterium alive in there and it produces carbon dioxide as a byproduct of its metabolism, it will distort the can because of the buildup of pressure. So you know that little dimple that's on the top of a home can something? If that's up, just write yourself a thank you note for whoever sent it to you, right? And then pour it down the sink. Because you don't want that, right? Like, you don't want honey that badly, do you? No, you don't. Okay, so that's botulism. And then black widow spider venom does something similar to these. It's a neurotoxin as well, but it's usually just local. So it's not really going to cause too much of a problem. I mean, it's going to suck, but. Okay, so the next thing that we're going to do, and we're not going to get too far into it today, is that we're going to talk about sort of a tour of neurotransmitters, right? There are at least 60 of them, and we're not going to do all 60. We are learning more all the time. As a matter of fact, we didn't even learn the neurotransmitter that is implicated in how marijuana affects the brain until 1992. So we're learning more all the time. We're not going to learn all 60. We're going to learn the ones that we understand the functionality of, or have become particularly relevant because of clinical applications. So I'm going to teach you some of them and not all of them. So let me show you what you have and then tell you that later on you're not going to be responsible for all of it. So there's a table right here at the end of this set of notes that has the neurotransmitter, the most common receptors, the chemical category, the function, and then some drugs and disorders. This is just here because people ask me these questions all the time, and I put it there for you so that the answers are there. Unless I talk about it specifically within the body of the notes, you're not responsible for it, okay? This is just because people are constantly asking me, okay, how does cocaine work? How does this work? How does that work? And I won't get to all of them in class, even though I'd like to, but they're in there for you guys. Okay, so how are we going to approach neurotransmitters if we're not just going to do a list of all 60 of them? Well, first we're going to talk about what we're talking about with neurotransmitters. You guys told me that you have to meet 2 criteria to be a neurotransmitter. What are they? They have to be released by what? Where? By a neuron where? Close to the receptor, exactly. These are the language of the nervous system. And so what I'm going to use is a chemical to communicate between either 2 neurons or a neuron and a muscle or gland cell. So if I refer to a neurotransmitter and I say that it's a CNS neurotransmitter, what I mean is it's communicating between 2 neurons in the central nervous system. If I say it's a PNS neurotransmitter, I mean it's communicating usually between a neuron and a muscle cell, or sometimes between 2 neurons, because there's a few that are communicating out here. But it's in the PNS. And a lot of neurotransmitters are both CNS and PNS. Okay? So sometimes I'll teach you the CNS function and the PNS function. So for instance, acetylcholine, its PNS function is, among other things, it stimulates all skeletal muscle contraction, right? PNS function. Its CNS function, I already told you one of them, learning, attention, and memory, short-term memory in the brain, right? Okay, so we will approach them and we'll talk about whether they're CNS or PNS. And so what they do quite often is bind to and open an ion channel, or ultimately cause the opening of ion channels, and therefore cause an EPSP or an IPSP. So if I use the term and I say this is a common excitatory CNS neurotransmitter, there's a lot of information there, isn't it? Right? Common excitatory CNS neurotransmitter. I mean, it probably what polarizes the cell, depolarizes the cell, and it's specifically usually being used between two neurons in the central nervous system. Does that make sense? Okay, so there are, like I said, approximately 60. We're finding more all the time. Neurotransmitter characteristics. They're not huge. We are not talking about huge proteins. Even the biggest ones are a few amino acids long, not hundreds of amino acids long. So that means that they're relatively low molecular weight. And I don't expect you to memorize looking at this. I just want you to look at a couple of the common neurotransmitters. Do you see what I mean by small? You can count all of the atoms, can't you? Right? This is not hundreds of amino acids long. I'm not talking about as small as a sodium ion, right? Most of them are relatively small. Here's another one. This is acetylcholine. Most of them are water soluble, not all of them. And this small water solubility is going to make diffusion across a microscopic synapse relatively quick. As they get bigger, they get slower with their diffusion across the synapse. So most are small and low molecular weight. They're relatively fast acting. And quite often there's more than one receptor for them. So conceptually, even though it's hard to understand, norepinephrine could be released and bind to a sodium channel in one area of the brain, and it would behave as an excitatory neurotransmitter. But it can be released and bind to a potassium channel in another area of the brain, which means it would be inhibitory there. So really, my question to you is it the neurotransmitter that really decides whether it's excitatory or inhibitory, or the what? It's the receptor, isn't it? Right? It's the receptor. It's basically the receptor that binds to the neurotransmitter. So although it's super annoying for students, once the brain learns how to make something, like acetylcholine, it can make it and release it as a chemical signal for a bunch of different reasons, right? That's why with physiology, when you try to just memorize what they're doing, you're not really going to do yourself a service. You have to look at what they're doing. So what is acetylcholine doing in skeletal muscle? It's binding to and opening a sodium channel, so it's excitatory. What is it doing here, right? Okay, all right. So the response to a neurotransmitter is tissue specific because typically a tissue has one or the other type of receptor on it. Is that okay? Okay, so how we are going to start doing neurotransmitters is we are going to sort of organize them chemically, but I'll just sort of show you, I'll give you a preview of things to come. So I'm not going to do this today because you guys are slightly ahead of the other class, but I'm going to tell you that when you look at neurotransmitters, you organize them by what they're derived from. Most of your neurotransmitters are derived from amino acids. So there are, for instance, the choline derivatives. There are these that are biogenic amines that are derived from a few different amino acids. There are these that are straight amino acids. And then there's some other ones. So although I know that chemistry is not always the most popular topic with you guys, we will start looking at the neurotransmitters and we will categorize them, and then we'll look at what they do, CNS or PNS function. And we're not going to learn all of them. We're going to learn the ones that you're interested in. And with each of them, if there is a disorder, schizophrenia, how does it relate to dopamine? Depression, how does it relate to serotonin? Then we'll throw in a few disorders and drugs just to make sure that we reinforce them. But that's not until after your test. Okay, I will see you guys on Wednesday. Yes, Wednesday. I will bring the Scantrons for you. It's part Scantron and part written. Yeah.